Good day everyone and welcome to part 1 of our revision lecture series on the basics of data representation in computational biology. As you all must know, I am Abhinav Jagtap, your instructor for this session, and today we will delve into the fundamentals of data representation in the context of computational biology. We have already seen this all things. In today's lecture, we are just going to revise things out. Before we begin, let me briefly introduce myself a bit more for those who have kind of forgot it. I have a background as a data scientist with experience working in the banking sector, and while I was working as a data scientist, currently I am serving as a faculty member for the university teaching department and CSVTU Bhilai. While my expertise lies in data science and artificial intelligence, today we are again kind of revising and emphasizing on the importance of effective data representation in computational biology, a field that plays a vital role in advancing scientific exploration, particularly in the life sciences. In computational biology, vast amount of information are generated, using techniques such as genomics. However, making sense of this data and extracting meaningful insights requires sophisticated computational tools and techniques. That is where data representation comes into play. As data scientists, we understand the significance of representing the data accurately. Proper data representation involves taking raw biological data, organizing it in a structured and visually comprehensible manner. Now, the significance of data visualization was the cornerstone of our discussion. Visualizations are powerful tools that enable researchers like me, like you, to extract hidden patterns or decipher complex relationships, communicate their finding effectively. We explored the diverse array of graphs and plots, each tailored to unveil specific aspects of biological data. In all this thing, Python emerged as the programming language of choice for our endeavors. Its open source nature and simplicity in terms of syntax, especially in terms of syntax, make it accessible to both seasoned programmers, like I will not say me, but there are some more seasoned programmers than me, and those new to the coding. You might be a now you might be a expert because you have already gone through a lot of video lectures. Python's extensive libraries, including pandas and NumPy, provide a solid foundation for data manipulation, organization, and statistical analysis. Now, these libraries are invaluable when dealing with the complex biological data sets. For genomic sequence, like from genomic sequence to protein structures, everything we can do with this Python. The hands-on implementation that we done was emphasized as the pinnacle of learning, while theories and concepts lay the foundation. Practical application cements our understanding, make a concrete base for us. Aspiring computational biologists and data enthusiasts were encouraged, like you all are encouraged to explore Python's capabilities, especially for data representation and visualization. Now, we also discussed the critical role of choosing right tools, in which we first of dealt with Anaconda, which is a comprehensive platform for data science, and it was introduced as a fundamental installation for the journey. Like we first of all kind of installed the Anaconda in the our PC, like CPUs, like PC or laptop, whatever you are having. Anaconda simplifies the installation of Python and data science packages, ensuring a smooth experience for all users regardless of their operating systems. The lecture that we initially saw also wrapped up with a detailed guide on setting up Jupyter Notebook. I hope you all remember how we set up the Jupyter Notebook, which is an interactive coding environment that encourages experimentation and creativity. It was highlighted that Jupyter Notebook goes beyond code. It facilitates the creation of visual narratives and breathe life into the raw data. So, with Python, Anaconda, Jupyter Notebook and at our disposal, the lecture concluded with an invitation to explore data types and variables, the cornerstone of coding, of course. These fundamental concepts served as building blocks for implementing data representation and visualization, especially in the realm of computational biology. So, in the next lecture, in the next step, we kind of delve deeper into the world of Python, focusing on its practical application in the field of computational biology. So, Python, with its open source nature, readability and extensive libraries, was presented as an ideal choice for this journey, though you have an option of using MATLAB and other languages. But the lecture began by emphasizing the importance of acquiring the right tools. Anaconda, a hub for data science tools and resources, was recommended for installation. The step-by-step guide provided that ensured that all participants, whether Windows enthusiast or Mac OS or even Linux, could set up the Anaconda without any hassle. You already know you have to just search it in a Google Anaconda, you just have to download Anaconda. From there you will get an option of download with Windows, you will just download it and just click next, whatever the pre-customized things you have, that will be more than enough. Once Anaconda was installed, we turned our attention to Anaconda Navigator, the gateway to the world of data manipulation, analysis and visualization tool. Within this hub, we encountered Jupyter Notebook, an interactive coding environment. I hope you remember Jupyter Notebook, which is an interactive coding environment that promotes experimentations and learning. Just go to open the Anaconda, open the Jupyter Notebook. The Jupyter Notebook in place, we proceeded to delve into foundational element of coding. The first thing that we learned was data types and variables. This concept were presented as crucial building blocks for our journey into data representation and visualization within the realm of computational biology. So, the data types in Python such as strings, integer, floats, booleans highlighted for the versatility that they are having. They empower developers to accurately represent diverse type of biological data, whether it involves gene sequences, numerical measurements or categorical informations. Variables akin to containers holding data were also introduced. Now, Python's dynamically typed nature, characterized by loose typing, was also explained, while Python automatically determines variable data types. based on the assigned values. This all the thing we already knew, that the moment I write 1.5, it will already take it as a float. Okay, we have a decimal in it, let's take it as a float. Python does this. This flexibility places responsibility on programmers to ensure data consistency and integrity as its flow through their code. The lecture kind of also concluded with the introduction of powerful constructions, array, list. The list is a versatile container capable of holding various type of items that were praised for preserving the inherent relationship between the elements, making them suitable for storing related values in biological data set. On the other hand, array tailored for numerical operations. See, I will tell you what exactly arrays are. So, for example, I have some similar data types. For example, I just want to take a list of students. What I will do? I will take an array, instead of declaring 10 variables, I will just take one array and it will do all the task of complete. So, they were introduced as indispensable tool for handling extensive data set effectively. This arrays outshine lists in terms of performance and memory utilization, especially when dealing with the substantial volumes of numerical data such as DNA sequence or protein structures. So, overall data types, variables, list, array served as main feature of our journey into data representation and visualization. They provide the essential building block to effectively manipulate, analyze, visualize complex biological data. We embarked on an exciting adventure into the world of bioinformatics and genetic analysis. The lecture was structured around captivating narrative of uncovering hidden secrets within the DNA of plants and finding potential cures for the common diseases using Python and BioPython. I hope you are kind of remembering everything. The lecture began with a recap of the power of data visualization in computational biology, emphasizing its role in the revealing hidden truths within the complex biological data. We revisited essential plotting techniques including scatter plots, histograms, the line charts. We also introduced matplotlib, a very powerful tool of Python for data visualization, as a valuable tool for creating informative visualizations. As we shifted our focus to bioinformatics, we also saw Python's BioPython library, and it was introduced as a powerful tool designed for tasks related to biology and computational biology. The SEQ object within BioPython was highlighted as a versatile tool for working with biological sequences including DNA and protein sequences. The lecture represented a practical scenario where Python and BioPython were used to detect the potential toxicity in the plant. You remember? Users who were guided through the process of collecting DNA sequences from plants and unknown toxic genes. A conditional statements was employed to compare the sequences and also provide warnings if toxic genes were detected within plant DNA. The lecture also kind of emphasized the real world application of bioinformatics, a field that combines biology and computer science to analyze and interpret biological data, particularly DNA and protein sequences. While the example provided was relatively simple, it showcased the power of programming and bioinformatics principle in genetic analysis. The practical tutorial on setting up BioPython was provided, ensuring that you, all participants, had the necessary tools for their bioinformatics journey. Then the installation of BioPython was compared to preparing for an adventurous journey, highlighting its importance as a comprehensive platform for biological data analysis. The lecture guided participants through the process of collecting DNA samples, converting them into BioPython SEQ object, sequence object, as you all have already seen it, and conditional statements to detect potential toxicity, whether a plant is toxic, whether this plant is toxic, potential toxicity in plant. The analogy of a DNA detective was used to make the process engaging and relatable. I hope you are all remembering this thing, that whatever I am telling. Next, we ventured into the field of biomedical research using Python and BioPython to search for cure for common cold that we all generally get. A hypothetical scenario was presented where a scientist aimed to decode a genetic sequence of the sniffle virus to develop a remedy. The lecture started by highlighting the importance of setting up laboratory, which in this context meant installing the BioPython library. This installation process was compared to gearing up for the scientific adventure. Participants were guided through the process of collecting DNA samples from different sniffle virus strains and creating a common genetic pattern by using these sequences. The BioPython SEQ object's versatility in manipulating DNA sequence was showcased. The lecture also introduced the concept of common genetic pattern, which needed to be identified to crack the genetic code of sniffle virus. A known antiviral gene sequence was presented as the potential cure, and the lecture culminated in a comparison between the antiviral gene and the common genetic pattern. The analogy of a scientific quest and DNA detective work was used to engage you as a participants and illustrate the practical application of Python and BioPython in your domain, that is biomedical research. Now, the concept of data integration was introduced as a crucial step in the computational biology. You were guided through the process of integrating diverse data sets such as genomic sequences, protein structures and gene expression data, this all to gain comprehensive insight into the biological systems. We also touched upon the significance of algorithms, if you remember, and also about the data mining techniques in computational biology. You were also introduced to the concept of sequence alignment, a fundamental technique for comparing DNA and protein sequence. We tackled the formidable challenges that computational biologists encounter in their pursuit of understanding life at a molecular level. Now, the lecture kind of aimed to prepare you as a participants for complexities and intricacies that might be faced in the field. We begin by discussing the ever expanding volume of biological data with advancement of technology. We are generating genomics, proteomics and many different things, many different type of data at an unprecedented rate. This data represents challenges in terms of storage, processing and analysis. We explored strategies for efficient data management and the role of cloud computing in handling large scale biological data set. Another critical challenge is the integration of heterogeneous data sources. Biological information comes from diverse platforms such as DNA sequence machines, microarrays. So, integrating these data sources to gain a comprehensive view of biological system require advanced data integration techniques and different type of standards. The lecture we kind of also touched upon the ethical consideration associated with the handling of sensitive biological data, emphasizing the importance of data privacy, informed consent and responsible research practices. The data is very new thing, especially in a country like India, but we have to have the data privacy rules intact. It is becoming increasingly essential for biologists to learn machine learning for various compelling reasons. The first reason being the data driven research. So, we have kind of seen the complete revision of classes that we have done. Now, I will tell you why you should learn the machine learning, artificial intelligence, data representation and things like this as a biologist. First thing is data driven research. In the field of biology, the volume of data generated through various experimental techniques such as genomics, proteomics and imaging has grown exponentially. Machine learning techniques are well suited to handle and analyze large data sets. Biologists can leverage these tools to extract meaningful insights from their data, identify patterns and make data driven decisions. Another thing is the complex biological systems that we have. Biological systems are actually complex with the numerous variables and interactions that are often non-linear. Machine learning algorithms, particularly those based on the deep learning, can model integrate relationships within biological systems. This can aid in understanding the complexity of biological phenomena such as disease mechanism, gene regulation and ecological dynamics. We can also create certain types of predictive modeling in which machine learning can be applied to predict various biological outcomes. Biologists can develop predictive models for drug responses, disease susceptibility, protein structure prediction and ecological changes. Such models can have significant implications in drug discovery, personalized medicine and conservation of biology or conservation biology we can say. We can also see some high throughput screening with the help of this. See, high throughput technique in biology generate vast amount of data in a short period. Machine learning can automate this analysis of automate the analysis of these data sets, helping researchers identify potential drug candidates, biomarkers or genetic variants associated with the disease in a more effective way. We can also kind of see some pattern or we can recognize patterns. Machine learning excels in recognition of subtle patterns in the data that might be challenging to identify using traditional statistical methods. Biologists can use this patterns recognize this pattern recognition capabilities to discover hidden relationships, anomalies, and novel biological mechanisms. We can also have some data integration with the help of this. Biologists are often need to integrate data from multiple sources such as genomics, of course proteomics, and clinical records. Now, this machine learning can facilitate the integration of heterogeneous data types, enabling comprehensive analysis and holistic understanding of biological systems. We can also have some biological image analysis. With this, we in the machine learning has very base for image analysis in biology. Computer vision algorithm can automatically identify and quantify cellular structures, tissues, and organisms in images. This is valuable for research in the field like pathology, neuroscience, and ecology. If you talk about personalized medicines, in this machine learning can enable the development of personalized treatment strategies by analyzing individual patient data, including genetic and clinical information. This approach has the potential to kind of change healthcare by tailoring the treatments of each patient's unique biology. Just imagine data mining and knowledge discovery. Machine learning can help biologists discover previously known relationships and hypotheses with their data. By applying data mining techniques, researchers can uncover valuable insights that may lead to groundbreaking discoveries. At the same time, we can also see certain kind of scientific collaborations. As interdisciplinary collaborations become more common, biologists who understand machine learning can effectively communicate and collaborate with computer scientists, data scientists, and engineers. This synergy can accelerate scientific progress and lead to innovative solutions of complex biological problems. We can also do some single cell analysis. Single cell sequencing technology have opened up a new frontiers in understanding cellular heterogeneity. Machine learning algorithms can analyze single cell data to identify distinct cell populations, track cell its cell paths, and uncover molecular mechanisms underlying cell differentiation, also their developments and disease progression, etc. One more thing that we can have protein structure prediction. Determining protein structures in a critical task in molecular biology. Machine learning, especially deep learning technique, has made significant strides in predicting protein structures accurately. This has implications for drug design, understanding protein interactions, and deciphering molecular functions. One more thing that I can think of is high dimensional data reduction. For example, high dimensional data, a common in biology, can be challenging to visualize and analyze. The machine learning offers dimensionality reductions such as PCA, principal component analysis, and T distributed stochastic neighbor embeddings. Don't worry about these terms. PCA and TSNE we generally call them, which helps biologists to visualize and interpret complex data in the lower dimensions. I cannot draw data with 4 or 5 dimensions in a 2-dimensional plane. I have to reduce the dimensions. We can also use natural language processing. We all know ChatGPT, right? So biologists can benefit from NLP techniques from extracting knowledge from scientific literatures, patents, and clinical records. NLP models can automate the extraction of relevant information, adding in literature reviews, hypothesis generation, and keeping up with the latest research findings. Now, Machine learning promotes transparency in research. By documenting and sharing codes and models, biologists can ensure that their analysis are understandable by peers, contributing to the credibility of scientific findings. We can have cross disciplinary collaborations. Just imagine, interdisciplinary research in the is very increasing, and it's very common. As biologists who understand machine learning can collaborate more effectively with computer scientists like me, or a data scientist like me, or the computer engineers. This collaboration can lead to innovative solutions and holistic approaches to complex biological questions. Having said this all, one thing that we have to keep in mind is the ethical considerations. Understanding machine learning also equips biologists with the knowledge to address ethical considerations related to the data privacy, bias, and fairness. As they work with the large data sets and make predictions with real world consequences, being aware of the ethical issue is very critical. Now, having this all things, we have proficiency. If you have the proficiency in machine learning, it enhances a biologist's career prospects. Many research positions and industry jobs now require or prefer candidates with computational and data analysis skills, including machine learning. Learning machine learning can open up broader range of career opportunities in academia, biotechnology, pharmaceuticals, and healthcare. We can also have some innovation and discovery, wherein machine learning can lead to discoveries by revealing unexpected insights and correlations in the data that might go unnoticed using traditional methods. Biologists who embrace machine learning can drive innovations by exploring new research directions and hypotheses. Biologists are often tasked with addressing global challenges such as pandemic, climate change, biodiversity loss. Machine learning can provide tools for modeling this prediction and impact of these challenges, and strategies for mitigation and adaptation. We can also have machine learning that can also be used to improve the quality of biological data through techniques like data cleaning, outlier detection, and imputation. Biologists can ensure their data sets are reliable, accurate, reducing the chances of drawing incorrect conclusions from flawed data. Machine learning can aid the data visualization, helping biologists present their finding more effectively. Data visualization techniques powered by machine learning can create interactive and informative visualizations or visual representation of complex biological data, making it easier for researchers to communicate their results and insights. Um, it can assist in experimental design by helping biologists optimize their experiments and sample sizes based on data driven insights. This ensures the resources are efficiently, and experiments are designed to yield meaningful results. In fields like ecology and environmental sciences, real time monitoring of data is crucial for tracking changes and responding to emerging challenges. Machine learning can provide continuous analysis of sensor data, enabling timely interventions in cases of ecological disturbance or environmental threats. By embracing machine learning, biologists have the potential to drive scientific innovation beyond traditional boundaries. They can explore novel research questions, create interdisciplinary collaborations, and pioneer groundbreaking approaches to biological research. In the face of global health challenges like infection disease outbreaks, machine learning can play a critical role in in the pandemic like situation, contact tracing, forecasting of spread of disease. Biologists are proficient in these techniques, can contribute to public health efforts and crisis management. So, having said this all, in closing, I urge you to carry the torch of knowledge determination, and ambition with unwavering resolve, your journey into the coding, data science, artificial intelligence, and machine learning is a noble one, and it will not only shape your careers but also the future of science and our world. So, go forth with pride, enthusiasm, and an insatiable hunger for knowledge. The world eagerly awaits the contributions that you, the future leaders in these fields, will make. You are the hope, the change makers, the torch bearers of the brighter data driven futures. Make it count. Thank you so much.